So what I'm doing just here, just update my toolbox. Oh, I shouldn't do that again. Okay, in the Teams channel, here is our agenda for today. We will talk about the CCA and using this MATLAB built-in function to look at the way to use this function. And there's theoretical tutorial about this CCA and this underlying principle in lineage algebra operation. The model, the method is called the generalized eigenvalue problems. Then we will look at a very popular algorithm called harmony. People use this for batch effects correction in single-cell data analysis. And this is the paper described harmony. It's published in 2019. It had been cited for thousands of times after that. Then we will look at this MATLAB function for dynamic time wrapping. It's very easy to use. It's a package in signaling toolbox. I don't know whether we have that installed. We can check it out. Then we will spend some time in Monaco 3, this package developed in R, and it had been incorporated in toolbox. So I will show you how to use that interface to get the pseudo time using Monaco 3 as additional variable. Then you can describe the states of your cells after that. Then we have some follow-up. Once you have this trajectory, you have this pseudo time, what you can do with the cells. At the end, we will discuss the highly variable gene analysis, and also if you have two data sets, you want to compare highly variable genes in between these two conditions of the cell you collected, and what you can use. And I will assign this at the end as a homework. I show you how to do this using example data set. Then your homework will be find your own data set and to compare and these two data sets using highly variable gene analysis, and then give the outputs of your detected gene sets to enricher or MATScape and see whether you can make some story out of it and see this comparison between two samples using comparative highly variable gene analysis in addition to differential expression analysis, see whether you can find some interesting discoveries using your own data sets. Okay, as we normally do, I just update my toolbox. So just make sure this is, oh, let's see, existing. Yeah, so I add this to the parse.
SD, right?
Yeah, it should be SC. I had a typo. I updated ST. Yeah, but this should be fine. It should be fine. Just make sure you have this latest version. I just double check. Oh. I can change it to this folder. Yeah, just make sure you have the latest version. This is version 24.3.9 with A. Okay, so if you have that in your interface, you are right. Okay, all right, yeah. So the function we will use is in this manual called external, because this function batch integration, the batch correction harmony was developed in Python. So we will call a Python version of this and get back the information. Well, the Monaco pseudo-time analysis is developed in R. So these two functions has been put under this manual here. So we need to know how to set up your R running environment. We will set up Python running environment. If you have done this before, you should have that package already. The environment should have already been set up. You haven't installed the Harmony or Monaco, but once you run it, the program will install it automatically. Okay, before we move to there, I want to see this CCA analysis. So let's using the documentation canonic correlation. So that's function CAN canonic. Yeah, so this is the function name for CCA. We start by looking at its documentation provided by the MATLAB statistic toolbox. So this is in statistics and the machine learning toolbox. So this section is different ways you can use this function, syntax. So you will provide two matrices, X and Y. And then if you ask two outputs, then the algorithm will give you two rotation matrices, A and B. So rotation matrix will be a squared matrix. And then whether it's a 2 by 2 or 3 by 3 or n by n, it depends on your input variable matrix. How many variables you use to describe your samples in X matrix, and how many in Y matrix. This X, Y can be two matrices with different dimensionality. So this could be n by 3, and Y could be m by 2. So it can be different, but you will project them and after rotation to the same lower dimensional space. And then the correlation between those projected points will be maximized. So that's the goal for CCA analysis. So we just use the example, the documentation provided. Just go through this function and look at its usage. So what you can do is just copy this section. It will create this example data, okay? I know you can open the live script, but we just do this step by step. Okay, so these two lines of code will read example data, okay, the car, bigger cars. And you have data by combining these variables. So this data set contains 406 car information with this. Each column is one of these variable describes this car's feature. And this data set contains missing data, so we have to remove those data points, those records, without the complete information. So for example here, the car number 15 has no information for the fifth variable, this MPG. So we have to somehow identify those rows and remove those to make a clean data set contains complete information for all the variables for all the cars. So this section of code is to, if you copy this, the function here is to identify whether the data at a particular row contains at least one missing value. If yes, this line will be removed. So only keep those rows with no missing values. And also, this data matrix has been split into two, into X and Y. X contains 1, 2, 3 variables, and Y contains 4 and 5, 2 variables. So this creates two matrices. One contains 3 columns, another contains 2 columns. So this is a 3D data, and also Y is 2D data. Okay, what you can do is just copy this and paste into here. Now we can close the data itself. This is original input data, but now we separate or clean the data by removing the missing values and also create X and Y. Okay, this is X, this is Y. If you use a scatter plot, so I put Y, the first column, and Y, the second column. So this Y is a 2D data. Yes, so we can look at the separation of the samples. Each point is a car, and described by X1 or Y1, Y2. These two. Look at their variability among them. You can also do this using X1, X2. If you only use two-dimension information, you can look at these two variables and the separation of the cell, the cars. And you can use scatter 3 because the data is 3D to look at. The relationship between these 3 variables and the distribution of these samples, those cars, in this 3D, 3 variable defined space. All right, so just imagine you have these 2 independent measures, X and Y. X using 3 variables and Y using very different 2 variables to describe. If you don't know the relationship between those records, for example, if you shuffle them, then you try to align this by the car's identity. So the algorithm you can use will be the CCA. So if you copy this, you will get several output. Okay, A will be a rotation matrix to help you to rotate your samples in X matrix. So X is a 3D, 3 variable, 3 columns, okay? So that means 3 variables to define these samples. So to rotate these samples, you need a 3 by 3 matrix. So A is a 3 by 3. But because it's truncated, so only use top 2, okay? You can, if you, this is like A rotate X. So A is a 3 by 2, X is N by 3. So this shouldn't work, but you can transpose this. Okay, this should work because if you consider the match of those inner dimensionality. So you apply the rotation matrix to X. This is projected after your rotation, and also reduce the dimension from 3 to 2. So this should be very similar to U matrix, okay? So A after rotation will become a U. Yeah, let me try. And the B is a rotation matrix for Y. So you can you can do the rotation yourself by Y multiplied by B. You will get this reduced dimension. And the algorithm will have already done this for you to rotate. So U and V will be the data point after rotation from X and Y correspondingly, respectively. The results of your own rotation, X rotated by A, looks not the same as a U. This because some of this internal manipulation, maybe the centralized data, I don't know, but in principle, this is the idea. Okay, so U and V will be the projected results. What we can do is to look at this projection of U and V and put in the same plot. So here I close all the figures I generated, but use a scatter 2, but U and V are 2Ds. So I will plot U first. And then I will type hold on because I'm ready to plot a V onto it. Semicolon at the end of. And this is from the first U matrix, then I plot V here. In different colors, so you start to see X and Y. X and Y originally and very different, but now after rotation, after projection, they overlapped onto each other. So our data set is special. They have one-to-one match between these two. So in theory, we should recover this one-to-one match. So I can give it a try, see whether this is index. What's the index for this? Yeah, so I should know what's one-to-one match between these two data sets. But there's not an easy way. There's no easy way to look at the correlation between them. What you can check is to look at this R, and this is a correlation coefficient, and to show you in this X and Y, these two reduced dimension variable direction, and what's the correlation between after you rotate the data. Okay, so this after aligned, that's your correlation coefficient. Oh, so you can check the R to look at how well these two samples were aligned. So 0.887, there's a correlation coefficient in that direction, X direction. So the first variable after dimension reduction. Whether these two samples aligned well. Okay, if perfect aligned, this correlation should be 1. This is in the second PC, basically, the alignment, how well they aligned together. Okay, so this is just some practice how to use this function. They're beautiful theory. You can look at this archive paper. If you click this, it will lead you to this PDF. And those are the place people put a preprint before publication and to share with the community. And this is a tutorial, basically this small review paper discussing eigenvalue problem and also generalized eigenvalue problem. So in CCA, the actually computation is to compute this so-called generalized eigenvalue problem to solve this problem, to get A and B matrix, rotation matrices. If you look at this more carefully, so here you have eigendecomposition equation here, okay? So that's the one we use to solve PCA, okay? So A is your covariance matrix. And then if you move down, it will have this generalized eigenvalue problems. Let's try to find the equation. So yeah, so this description is so-called generalized eigenvalue problem. So here you have another matrix B, okay? You consider A is from X, so one data set, and then you get the covariance matrix and it become A, and then you do this for Y matrix, and then you do the covariance matrix and become B, and then you solve this eigenvalue problem. The solution will provide the rotation matrix for A and B, and those corresponds to X and Y, original. Okay, so that's if you can solve your problem and translate your problem into linear algebra operations, and basically you translate your problem into matrix multiplication problem. and then you use some of this eigen decomposition. It will be very elegant solution if you can solve your problem in this way. You probably just borrow those ideas and then modified how you define this data matrix, what's your goal, and translate those problems into linear algebra operations. If you cannot do this, normally data scientists will push those kind of problems given to neural network. This is another level, and to deal with more generalized problem and involve nonlinear components, you'll still be able to do it. So that's a general trend in the field. If you cannot solve this, use basic linear algebra operation combinations, then you give the problem to neural network, use GPU power to compute those parallel operations. Okay. I think we are going to move to another topic. This is a very popular batch effects correction algorithm. We can start by looking at its paper. So this is published in Nature Methods in 2019 and has been cited since then more than 2,000 times. So many studies will run this or copy others' publications too, because other people also do this. So I believe most of users do not ask why they try to do this. I would say it's very much useless. I don't know. But we just show this, and I still have that function in the toolbox, but I don't suggest you to use it. Okay. I probably will remove that from the toolbox. Okay, this is their picture one, figure 1 to explain what they try to do. You have 3 data sets color-coded in 3 different colors. And then you run this clustering algorithms and also have a cell type defined. Well, I don't know whether they are defined or not. Maybe they using these gray colors because they don't know the cell types, but they know there are 3 different cell types in there. And those 3 different cell types was indicated in this different shape. And the same cell type, like this funny shape, should be clustered together, but because of data sets from different sources or different treatment conditions, then you have subclusters for this particular given cell type. And some of these subclusters and even close to another different cell types. Okay, this creates some confusion whether you should, if you have no information about the cell type, you may look at, okay, here is a cluster, maybe same cell type, but 2 subtypes. Maybe you cluster the way differently from this ground truth. Here they assume they know all this. So the idea is you have some data set looks like this. You want to identify those focal points and then to start to move the cells based on the nearest neighbors to change its coordinates in the embedding a little bit to make them similar ones more closer to each other. And rearrange cells. And then at the end, after the several iteration, you reach some levels of this harmonic states. The similar cells of the same cell type although from a different data sets should be moved together. Okay, so this makes people feel better, and from your experiment you don't have any systemic shift between data sets, and the same cell type has already been put together. But this operation does not change the UMI count matrix. You still have your 3 UMI count matrix. You just change their embedding coordinates. So you use t-SNE, UMAP, whatever, and have this embedded position. So if you consider this as a t-SNE, t-SNE 1, 2, so you have those embedding in the 2D scatter plot. So you just move cells by their neighbor relationship and try to avoid the separation of data sets. That's all the program is doing. Okay, come back to the toolbox here. So I clean my space. We need to make sure we have Python installed. So if you type mini conda. So if you type mini, if you have already installed this Anaconda environment, you should have that in there. That's all we need. If you use a new computer or log in with different accounts, then you probably don't have this installed. Then you have to download the mini conda and install it. Because this environment contains Python, so we can start here and see which Python we should have that. Yeah, so it just showed me where I installed my Python. So this information will be used when we set up the Python environment in MATLAB. So we just have to point our parse to this place that MATLAB knows where Python was installed, which version of Python you want to use. You can have multiple Python, even different versions installed on the same machine. But here we just use minicodon provided Python, and the one is right here. Okay, for those who came late today, make sure you update your package. Okay, your latest package should show this version number. Okay. If not, you please update. So this version is 2439A. Okay. So to set up a Python environment, you have to first click this external and then set up a Python environment. Then this dialog window will indicate where you have selected your Python version. So the place is consistent with what I just showed. If this is not correct, please just use another to search for the parse and select Python. All right, should we move on or just give 1 more minute to set up? Everybody is fine? Okay. So I just load my example data, okay? So I just load samples processed. And then using this label, I will look at the batch. So now I'm going to do the harmony to align these two data, to move them, basically just move them, overlap onto each other. So the first time run, so you just select this, and I hope everything works. This gave you the description, the PubMed ID if you want to read more about this. If you're ready, just continue to run this harmony. Yes. Use this. I believe this is the first time run harmony, so the program have to download the harmony and install on this machine. So this will take some time. But just give it a try. And if it's not run through, we just let it go. And this is not important. I don't suggest you to use this. Okay. So in my case, this is updated. So the cells has been moved around and make them, so updated these embeddings. So let you to feel comfortable. If you are not happy, and you can run that again, okay? I just run this one more time. It continues to do this optimization. Use this. The cells will be moved further and maybe even closer to each other until you feel comfortable. Okay, seems we correct the batch effects. I suggest a different strategy. So I use this over clustering and annotate the cell type independently. If they are same cell type, we just merge, compare the same cell type, even the same cell type cells from two different batches of data. They separated, but they are still same cell type. I'm not moving their positions. Okay, but can you everybody successfully run this? Okay, but that's all. And if you are interested in, you can read the original paper and maybe read the citing paper and use these tools and see whether other people use harmony to do anything more meaningful. But to me, I still don't understand the logic of doing this. Maybe it's to facilitate the annotation of cell types. Okay, now we move to another topic. This is about the dynamic time wrapping. We just play with that function. So I can temporarily shut this down, clean my space. Yeah, first let's see which I may not have this installed. Oh, good. So on my computer, it's installed in the toolbox called the Signal. This is Signal Processing Toolbox. You have the function DTW under this folder. So we start from looking, its documentation. So this is a distance between signals using dynamic time warping. So you provide two time series data, and then if you directly compute the difference between these two signals, because they have a different sampling frequency, one may sample very sparse across time, another maybe very frequent sample. You get the signal with different lengths and different sampling density, and the data points between these two signals not matched. So if you compute the distance between these two vectors, the distance will be huge. But if you run this alignment first, you get two aligned signal, Ix, Iy. In that case, the distance is more realistic after you really stretch the length or to match the points internally. So the sample they provided is a simple cosine this shape. So what we can do is just copy this too, okay? Copy this line to create X and Y, two signals. So X contains 1,000 data points, and Y has only 399. If you plot X, okay, it looks like this. And you can see plot line and the dots. Okay, so in this case, you have this smooth curve, and also you show the points here. Okay, there are 1,000 points, index from 1 to 1,000. We can allow this plot to hold, so let's hold on. Then you can start to plot the Y using, for example, square. It will give you using different colors anyway, but you can also add the different symbols to markers to show you the difference. So you have two time series signals, then there are some circulation among them internally. So now we are going to align these two before we compute the distance between these two vectors. Dynamic time warping X and Y. Yeah, we just show this. So the answer is 21.3. So this is the distance provided. If you put a semicolon at the end, see, I don't want any outputs. Just I think this program. Yeah, this program just generate this figure automatically for you, the whole thing. That's all. So you have original signal in the top panel, and then you have aligned signal. Try to minimize the distance between data points overall. If you really need those points. So you have a D, the distance, you have Ix and Iy. So those Ix and Iy are the aligned points. You can plot Ix, hold on, and then plot Iy. In a new figure. So you can certainly plot yourself the aligned signals. Oh. Let's see what's in the Ix. Oh, Ix and Iy is just index, not the original signals. So we don't plot in this way. There might be the index. So I can put X here and change, and Y using its index to reorder. Yeah, I just gave it a try. This is aligned. Okay, so rather than plot the index, but you resample your original signals, use this index. The index will have one-to-one match to align the two signals. As I said, I haven't found the application of a TWD in single cell.
Here we are plotting the values of X in the position Ix.
Yeah. So the Ix is just 1, 2, 3, and the corresponding maybe 2, 4, 5. So try to align after the two signals. Yeah, so it has been applied in some of those trajectory analysis internally. So there's some steps, maybe you need a dynamic time wrapping if you have, maybe through the comparison of the two pseudo-time trajectories, okay? Maybe you can have do this comparative pseudo-time analysis. You have one sample and then you believe another sample, the trajectory should align with the first sample. Here we have, indeed, there's one publication using TDW to compute the TDW score to combine the time series data, enable predict gene change. Yeah. Yeah, basically every machine learning algorithm has found applications in single cell data. If you Google search, this one is one of the example. They have time series single cell data, probably want to align some of those to match the signals. Okay, any questions for this part? No? Now we are moving to trajectory analysis. So we have to make sure we have R installed. So here we have to see type R Studio. So in my case, I have that installed. So R Studio normally will install R at the same time as a package. So at least in our university system here, we install R Studio will normally automatically install R at the same time. So you can double check you have R Studio installed and also the version of R installed once you start the R Studio. Then if you click this link I provided next to the Monaco 3, you will find Monaco 3's website. This version 3 has not been published, so if you want to use, you may cite Monaco 1 paper, Monaco 2 papers. So Monaco 3 try to become a comprehensive package like Surat. Not only do this pseudo-time analysis, is try to increase the functionality by providing more functions, for example, for clustering, for differential expression. If you are R user, you should be able to follow the instruction here by something like getting started with Monaco 3. Okay, to do this, there are subsections to do trajectory analysis. So that's the way you put you read your data in and create objects, and then process, do dimensionality reduction, and create the U-map plot like this. And then that's for single cell only, yeah. In this case, you can do the feature plot to show those selected genes expression among the cells. And then you have a cluster function to do this clustering. Then here you start to run trajectory to learn graph. Order the cell with the pseudo time. They provide an interface here to allow you to select the root nodes. So after the create this graph, you know, okay, in the center of this cluster, I have a root, and most of other cells are developed from this, start from here. So just give some direction of your trajectory. You can select any cells. You can select, see, this part as a root if you believe that's true. Then the algorithm will start from the root and start to give assign a pseudo time for all the cells in this plot, because you select the center here as a root. So the cells close to this center will have a smaller pseudo time, and after you move away from the center, you get larger pseudo times. You have early stage and later stage, basically like that. Okay. And then you have other operations too. Select parse. You can even do the 3D trajectory. So in general, this is a very decent package. So if you want to use original version, that's the place you can follow the command lines and give you some very detailed instructions. Okay, I think we need to take a 5 minutes break. I come back and show you using this interface, we can access most of those functions using MATLAB without even know those command lines. Okay? Now we are going to see how can we using Monaco 3 without leaving the toolbox. So I will load example data. Process. So this data set contains many different cell types. So I want to simplify the data type in order to make the job for pseudo time easier. Okay. Let's see. I look at the cell type, and most of them, if I highlight this light blue, this is alpha cell. So we have a lot of alpha cells. So I will extract alpha cells. So what you can do, just click this button here. And this will allow you to select cells by cell type. And then you have this separate view for different cell types. And then you click this finger button to extract one of the cells. Cell type, the alpha cell is on the top because this contains most of cells. You select this only alpha cells, then click OK. So this view contains only alpha cells, so you can close the other two. Now, because this is a subset of cells, you probably want to do this QC again to remove genes has no expression. What I will do here is to remove mitochondrial genes. Just make the search space smaller. Remove ribosome genes. Okay, remove the genes without approved symbols. That's mouse data. Okay, every cell, every gene is fine. And then I do the basic QC. Stringent. So those operations just make the data cleaner, okay? Because there are so many genes after you extract those cells from a large population. Those genes may not express in this particular cell type. They express in other cell types. So contains those rows with no expression doesn't make any sense. So now we have this relatively clean data set. We are trying to do the trajectory analysis with this. So now I can select set up R environment. Or maybe I save this so I can load it back next time if I need. I can export to workspace. Because I have done so many operations, I want to save my temporary data set here. Once, if I have something crashed or stop running here, I can still load back this view. Okay, with this, we can start to check our running environment. So this point me that where I should call this R executable file. So this has been set up on my machine previously, so I have this. Otherwise, you have to select use which R version and where you installed it. Normally, on those machines in the university, you should have R installed in C program files, R, and different versions of R. Inside this version, there's a bin, the binary file, and within that, you have R script. So you select that and give that information to the program, and it should be all right set up. Okay. Now I can just click pseudo time analysis. Then it will ask a working folder, so you can use to save your temporary intermediate files. So now this tool tells me I need to brush a cell to include this as a root, because in the middle of the analysis model 3, it requires you to point a cell which this is a root cell. So that information is required. So we have to find a place. So I just assume this is my root file. I can brush this. Okay, point. If you brushed more than one cell, it's okay. One of the cells will select as a root. So now, after selecting this root, I'm ready to run pseudo time and use this as a working folder. Oh, I use is a brush.
No, like what does it do? Like selecting that bunch of cells.
You can select any cells you believe to be the root. I have no.
So as a root, where did it start?
Where did it start? Yeah. That's right. Use this working folder. You can generate 3D, keep temporary working files. Yes. Okay. Again, this is the first time I run this. The program will download and install Model Code 3. This first time will take some time to install and run. You can see it's busy behind this window. Okay, it's trying to install the packages. They should never seen this before.
Isn't that there's an error in one of the packages VIBS manager?
Oh, then I have nothing to do with if that's the case. Yeah, so it's still working on that. I don't know. But after that pop-up window, I just select ignore it maybe. So the program just try to download a lot of dependence. Yeah, same problem here. Yeah, let's see you again. Well, yeah. Yeah, so you click this button here, yeah. Then say yes, you want to do that. Then cell type. Mm-hmm. Okay. No? Yeah. Mm-hmm. Then it will pop up this. Then you select this button here. And then, yes. Then you extract. Yeah, so, yeah. Yeah, so the problem. Yeah, if you see, okay, same problem. Well, yeah. Yeah, so we're running into the same problem. I have to update this. Let me think. Yeah, so we cannot do this today. There's one problem in the middle of the running. There's one variable has not been assigned. Yeah. But after I fix that bug, you should be able to. Yeah, maybe I just do this right now. Mm-hmm. You have different errors. Yeah. File manager is not installed. Yeah. Yeah. Can you? Yes. Maybe just click. Try another one. Yeah, I think you are right. You just select this. Yeah.
That's what I
Uh,
have selected on.
It doesn't matter anyway. So this package right now has a while this problem not assigned run Monaco 3 function. So yes, let me see what I can do with this. Yeah, so there's a problem in the middle. So I have to spend some time to fix this this bug. Before you can actually run this, okay? So if you finish that, then the program should give you each cell is pseudo time. And then you should be able to select here and so you should have one more variable, see, pseudo time. And then to give each cell a different color. Okay? In this case, this is not a pseudo time, it's something else. But you will get something similar like this. So what you can do with this pseudo time is to export your data, okay, into the workspace, and to get this pseudo time information and to do regression for your genes. Remember, we had tried lasso. Lasso is one of the methods you can use. You will give to do this kind of regression, you give X, this is the expression matrix, and your Y will be the cell with its pseudo time values. So this algorithm will pick those genes associated with Y and to show you the top ones if you select the top. Okay, just refer back to the regression lecture. You should be able to find those solutions. So because of this bug here, so I'm going to show you the different solution to do the pseudo time analysis. So we have alpha. Then I'm going to use fate to do the embedding. So I can run 3D fate, okay? As I explained, fate is based on MDS. So the algorithm is assumed that you can keep the distance relationship between cells, even the distance between them in high dimensional space is large. Well, t-SNE will ignore if you have two cells, they are too far away from each other. It will consider those cells are not neighbors. So when it regenerates the map, it will be randomly put this cell here and another cell somewhere else, not close cluster together. Well, the fate will take that information, try to keep the distance same as its high dimension space because of using MDS, multidimensional scaling. So that's why I recommend to use fate to do the embedding before you run the way we are doing this pseudo time. So why is it so slow? All right, so this is my fate results. I don't know whether you get the similar thing or not. I can wait. I will clean up a little bit because I'm going to build the trajectory, and I don't want to cells in this region to drag my algorithm towards that outer layer. So I will delete those cells. I will delete some of those cells. A brush. Maybe I shouldn't delete those cells. Those cells may be important to build the bridge between them. I don't know. Anyway. So I will make this clean. I just try to make the algorithm job easier, okay? Because there's obviously some of the trajectory in this plot here. So I try to make this clean. So after remove those cells, you want to apply QC again because the expression of some of the genes because of this operation will change. So we want to get rid of some of these genes contains no expression information. Now I want to save this in my workspace temporarily in order to get it back when needed. Okay, with this embedding, and I'm happy to start with analysis. Where is my, using my plot, cell trajectory. You can try both. So let's try this first. So you can see it start to create this so-called spline fate curve. And then you see, yes, you want to select those highly variable genes using Spearman correlation to select several genes which changes expression across this trajectory. So this function can be refined later, okay, if you need this kind of analysis. For now, it's just a demonstration. You can get some of the genes without even use lasso or other method to export your data. But in general, we can export this pseudo time and into a variable y and use lasso or regression to find, not through the interface, but the interface right now is only for demonstration. You can do to find some of the genes. And so this is one way. And if you look at this, you should have your spleen fit pseudo time. Okay. If as I said, if you can run monocode correctly after I fix the bug, you should have one more variable called the monocode pseudo time. But now we have the spleen fit pseudo time. You can color code your cells based on this pseudo time we just constructed. Okay, this end is early stage and then become later stage. You can change the color code. You can change to different color map. Okay, basically this is like a trajectory. As I said, you can also use principal curve. Sometimes the principal curve give you better resolution, better results, but not in this case. Okay, this principal curve gave me this trajectory like this. But sometimes it gives even better fitting. Okay, so I'm not going to use principal curve results. Spleen fit is better. Again, this is not the best fitting. The reason is, although they are alpha cells, also I already cleaned very much, but they from two different batches. If you look at the batch ID, okay, one from one batch, another from two batches. But if you only work on one of these batches of data and to have this trajectory, this could be very useful. So what I can do is just remove one of the batches. I can select cells by batch ID and select, for example, only the red cells. So now I have an even cleaner data set. I do QC removes 800 cells, genes. So in this case, both algorithms should be get the similar results. Yeah, so in this case, I create create those pseudo time for those cells, and the result may be compacted by some of these outliers. So you can make a better, you can delete those cells. For example, you remove some of the cells, you can still have this trajectory and then assign cell pseudo time. If you believe those alpha cells, although from same time point, there are some more differentiated and more matured, so you have those information, and you can start to use that information to get genes or gene programs, see what are the gradual change from one end to another. Okay? Make sense or not? So I don't trust more complicated or more complex structure of the cell population. But for the simple ones, I believe you align them, this could be true. But if you have multiple branches, I don't think algorithm will create very beautiful results. So my strategy is to extract the cell population, and you know they are at a different stage. And then there's very simple structure, and you know the gradients. Okay, any questions? Okay. So I can just keep using these examples to show the highly variable gene expression. Okay. As I said, highly variable gene expression analysis will require a homogeneous population of cells. So you don't want to have two populations. So in this case, we have a clean data set here. So only one cell type is alpha cells from one sample. And we have already removed quite a lot of these outlier points. Okay, still keep doing this. And then you can click this button to see highly variable gene analysis. So all the cells, the spleen feed method. Okay, we want to identify those genes which jump out from the spleen feed curve. Our hypothesis is those genes are the most important feature genes to reflect the activities of those cell types. Okay. Okay, so the analysis will help you to identify those genes by looking at the distance between genes to this curve. So you can select top 200 or 150. so-called highly variable genes, then you can send those genes to a table to select these genes. It's this here. Now with those genes, you can send those genes to enrichment analysis, which is this button here. That's the page for enricher analysis. The question was, as a group, what those highly variable genes are functionally enriched for. So you submit this against a background, 150 genes. Okay, so this is from this analysis for those group alpha cells. So now, so your homework, this will be part of your homework to find a cell type you want to study and download the data, clean the data, to run this single sample highly variable gene analysis and identify those featured genes, highly variable genes, and send those genes to enricher, okay, to annotate, to discuss the function of those genes involved in. Another will be comparative analysis. For example, in this case, we have highly variable genes analysis you can apply to blue cells and the red cells separately. But we want to compare the results. So this is called differential variability analysis, or DV analysis. The method we just finalized. So this analysis will apply the highly variable gene detection for both cells, both set of cells separately, and compare, see which cell changed mostly between these two samples. The change, we mean the deviation from the regression curve changed most. So which one was, for example, on the regression curve before, but after, in condition 2, this gene jump away from the curve. And it will be, we call it variability difference, and we want to see which gene the variability is very different between these two samples. So again, your homework is to find a data set contains two conditions to compare. Then you have to using homogeneous population of cells, okay? You have to compare alpha cell against alpha cell. You don't want to compare between different cell types. And then the comparison between these two cell types, batch of data indicate that different development time. So if you read the experiment that generated these two data sets, they are mouse samples from a mouse development in different weeks. So our comparison will identify the differentially variability genes across these two samples. So once you prepared your cells, you click this button here, and then to continue, you want to compare between batches. Okay, if you select cell type, this should no difference cell type. They're all same cell type, so that's invalid selection. You want to compare between two batches. That's between these two, okay? Select OK. The program will run highly variable gene detection independently and then compare the difference and give you the output for the table. So the table should contain the results. That's all you need, but you can also continue to show the difference, okay? So the table is here. So this plot is the same as the curve you just see from the single sample analysis, but now we have two samples. One is blue, one is red, and you have two curves. One is blue, one is red, because we are doing the contrast to compare them. So you can highlight one of the genes. For instance, this gene called SST. SST, because it's the same set of genes, but you have two conditions. So the same gene will appear twice in this figure. One corresponding to blue curve, another corresponds to red curve. But the distance between them for this particular gene is not large, so its relative position very similar. And this also reflects in this profile in blue cell and the red cell. They are similar. So no major change. That's not the gene we want to detect. We want to detect a gene which looks very different. Okay, this is in condition 1. This is in condition 2. This B2D, I just clicked by chance and found this gene. You can try different, so no difference between this gene, between two conditions, but there's some changes here, okay? It expresses 20% of cell in blue cells, but 60% in red cells. So that's created a major change. So we want to identify those genes have major change. We believe those are the important genes has been regulated because of the process of this development time. Okay. Depends on your study. If you study the disease, the healthy control, then you have a different interpretation of the genes change greatly. So you want to identify those genes and create the list and to see whether it makes sense to tell you the difference between two samples. Yeah, so that table is ranked. So the table will tell you the most different ones on the top. Okay, B2B is one. I just by chance click that. This is number 4, the most different ones. CD74 should be the one, so I can select the CD74, light it to highlight. This is a huge change, okay? So you want to look at this table after you finish analysis. This table, look at those genes, see whether those change of the expression profile will tell you the difference between samples. All right, that's the homework. I will document what I just said in the homework description. Oh, that's a good question. So right now you have only pairwise comparison. If you have 3 samples, you have to compare 1, 2 first, 2, 3 first, 1, 3. So you create this 3 ways, multi-ways comparisons. Yeah. You can, if you want to group 2 and 3 into 1 and compare with 1, you just rename the labels, batch ID. If you rename the batch ID, like batches 2 and 3, rename as a batch 23 and batch 23, then the cell will merge as a one group. Yeah, this is interesting for alpha cell in this mouse pancreatite. So this is week 8, and this is week 14. In week 8, there's only 6% alpha cell express CD74, but after several weeks, you see 98 of the alpha cell express this, and created a huge difference. Yeah, whoever study alpha cells probably know what's happening here. All right, just let me know what you if you have any questions. But I will go back and fix the pseudo time bug. It's very easy to fix. Maybe just take me 5 to 10 minutes, and I will let you know. And then you upgrade your package and try at home if you like. Okay. Yeah. All right. See you next week, everyone.